Good afternoon, everyone. I'm Jane McIntosh. I'm the Chief Philanthropy and External Affairs Officer at the Alzheimer's Drug Discovery Foundation. Thank you so much for joining us today. I'm here with Dr. Chief Science Officer at the ADDF. And today is July 1st. We are halfway through the year, and we are going to spend the next half an hour or so talking about some of the incredible advances that have happened within the Alzheimer's research field in the last 6 months. Howard often likes to say this is harvest time, and we have a lot of interesting developments to talk about today. So, Howard, how are you feeling about this first half of 2024?
Yeah, it's been really amazing. Like you said, it's harvest time. The reason I say that is we've had research on Alzheimer's for about 45 years, but during this year we had the second disease-modifying agent really come to an FDA committee with the unanimous advice that the donanemab from Lilly was efficacious and that there was a substantial benefit to risk ratio, a positive benefit to risk ratio. So this is really the second disease-modifying agent to be approved, which happened in 2024. And we also had some major advances in diagnostics, including a licensing deal, which we'll talk about in a few minutes.
Yes, it's all very exciting. Let's talk a little bit, go into much more depth about donanemab. From your perspective, I remember when I first started at the ADDF, Aduhelm had just gotten approved. So we're now 3 years later. Can you talk about the drug and also talk about what was specifically different about the clinical trial that Lilly designed?
Sure. Well, Lilly's trial did some innovative things that helped advance the field. One is that they kind of used the Goldilocks strategy to enroll patients into the trial by doing tau imaging. And tau imaging is important because it measures the tangles in the brain, which are characteristic of the disease and indicate neurodegeneration. And the idea in using tau imaging was to identify people who were not too early in the disease and had so little pathology that it would be hard to imagine that we would be able to see a clinical benefit from treatment. And on the other side, not enrolling patients that had too much neurodegeneration, too much tau imaging, because it was thought that if there was too much, that it would be hard to show a beneficial effect of donanemab, of the drug. So that was one thing. It turns out that it's likely that using tau imaging in clinical practice at this point in time is not going to be practical, but it is possible for clinicians to identify people in that mild cognitive impairment, early dementia stage, just based on a clinical assessment without the tau imaging. So the tau imaging probably helped Lilly to really define a good therapeutic window. And the other thing is that Lilly introduced into their trials was a stopping rule, essentially, that the PET scan, the PET amyloid scan that was required for entry into the trial to demonstrate the presence of the target of the drug that Lilly was testing, is also that they did a follow-up PET scan at 6 months, 12 months, and 18 months. And if those follow-up scans showed a complete removal of the amyloid from the brain, it was reasoned that there was no reason for further treatment. And so having that stopping rule in clinical practice will be very important because there is a burden of getting these drugs because they require infusions. Eisai Biogen's drug requires 2 infusions a month. Donanemab, Lilly's drug, requires 1 infusion a month. But either way, the infusions are somewhat burdensome. And we're looking forward to a day when, not too distant future, when these drugs will be administered at home through a subcutaneous pen, sort of like the way people take insulin these days. So that was, sort of in summary, I think that kind of what the committee considered in the donanemab evaluation by the FDA commission. And the FDA ad hoc committee gave a unanimous decision both on efficacy, which was one question the FDA asked the committee, was 11-0, and on the benefit to risk ratio, where the committee voted 11-0, that there was a therapeutic benefit that outweighed the risks.
And you've often said, Howard, that while this is, an amazing step within advancing uh, Alzheimer's treatment, that there is going to be uh, combination therapies really is the future of really treatment for this disease. Can you talk a bit about combination trials and how the ADDF is approaching this work?
Sure. Well, like cancer, we think that combination trials, combination therapy will be necessary to effectively treat the disease. If you think about the fact that the donanemab and the Leqembi from Eisai Biogen slow it down about 35%, somewhere in that range. We want to get to, that's a modest benefit. We want to get to ultimately, let's say 100%, or even preventing the disease itself. And it turns out that what we're seeing is that we're going to need, how do we get from that 35%, which is kind of the benefit that the current drugs show, to the 100%? And it's generally thought that we have to use combination therapy to attack the other mechanisms that are involved, like inflammation and epigenetics, which is sort of genetic modifiers that occur with aging, basically translating the biology of aging, because aging is the leading risk factor for Alzheimer's disease. And there are many mechanisms with aging that affect the brain, cause neurodegeneration. There's about 7 or 8 main pathways that the ADDF is investing in. I'll give you one statistic that I think illustrates what's going on. About 5 and certainly 10 years ago, about 75% of the drugs that were in clinical development were targeted against the amyloid and the tau pathology. So we were targeting the pathology. Now the field has almost completely shifted to what's the biology that's triggering this pathology, that's resulting in the pathology. And again, now 75% of the drugs in clinical development are targeting these mechanisms that we would say really relate in some ways to the fundamental mechanisms of the biology of aging. So non-amyloid, non-tau drugs are the future of the therapeutic development, and they'll be used in combination with these anti-amyloid monoclonal antibodies that remove the pathology and the downstream effects of the pathology on the brain.
Howard, can you give an example of a company that we are currently invested with that's sort of an exemplary combination trial?
Well, we recently invested in a company called Coya Therapeutics, COYA, and they have a combination therapy that involves 2 immunological agents, because for a long time, the immune system was recognized as probably playing a role, even going back to the days when Alzheimer first described the disease. He recognized that there were inflammatory cells around the plaques. And we haven't had a way to attack these inflammatory cells, because while they're beneficial, these are the cells that remove the plaques from the brain. They can also be detrimental, because by causing too much inflammation, these cells can actually be damaging. And so what Coya Therapeutics is doing is having a 2-pronged approach to the immune aspects of the disease, as a way of controlling the inflammation while making sure that the part of the cells of these immune cells that are attacking the plaques are more effective, and there's more of them. So Coya has a dual approach. And I might add that even the trials that it's interesting that even the trials that use the monoclonal antibodies like donanemab and Leqembi were actually combination therapy studies, in the sense that people were on baseline therapy involving current drugs on the market, like donepezil and rivastigmine and so on, because these cholinesterase inhibitors are generally used in practice. So in a way, the trials that go on, just by their very nature, are combination therapies. What the ADDF wants to do in its advanced combination therapy clinical models of development find more efficient ways of doing these studies, because they can be very large and expensive, and we want to take shots on goal with combination therapy. So one of the things that we're doing is looking at ways to better conduct combination therapies in a more effective way.
So Howard, another area of focus for the ADDF is biomarkers. And you've said many times this year that 2024 is the year of blood test. Can you comment on the news that we heard this past week from C2N?
Yeah, well, about 15 years ago, we made an investment in a company called C2N, which was spun out of Washington University in St. Louis. And they had a very novel and have a very novel approach to a blood test using a technology called mass spectrometry. And this is a very sensitive and precise way of measuring things in the blood. And ultimately, the company has developed their test to the point where the test is now on the market and available to doctors in 49 states throughout the United States. There's only one more state that we're waiting for approval, and that is in New York, where we have a very strict regulatory environment for approving blood tests. But imagine there's a world today where a person can come into their doctor's office with a memory complaint, perhaps get some brief memory testing, want to know if they have Alzheimer's disease. The doctor can order a simple blood test, and within a reasonable period of time, let's say a week or two, whenever the C2N results come back, can identify with over 80%, close almost to 90% certainty, to predict what's actually through the blood test going on in the brain, whether there are amyloid plaques in the brain, for example. And so this test, which is now called Precivity's AD2, has been shown to be a very effective test. And it really illustrates the new world of diagnosis for Alzheimer's disease. But the test will also change the way we do clinical trials, because up to half the cost of conducting a clinical trial is finding the right patients. And some of these phase 3 trials are $400 million. And so reducing that $200 million of finding the patients is going to be a big breakthrough. Another test that's recently been approved, that recently had some success, was along the same lines through a company that we invested in called ALZpath. And this test is more of an immune test. And they have a monoclonal antibody against the same target, which was this p-tau217, which is a specific chemical part of the tau molecule that makes up the synapses and the connecting cables between the neurons in the brain. And ALZpath just did a licensing deal with Roche Diagnostics, which is probably the largest diagnostic company in the world, with over 70,000 placements of their machines throughout the world. So Roche wants to use the ALZpath antibody to incorporate that into their technology, their diagnostic technology, which is, again, another big breakthrough, because that test may even be lower cost and hopefully just as precise and as easy to administer. So we have 2 blood tests that are on the market or coming to the market in the near future, which will really, I think, change clinical practice, diagnosis of Alzheimer's disease, and the way we do clinical trials.
You know, Howard, it strikes me that when you are speaking at different symposiums and you talk about the blood test, often people will raise their hands and say, but how do I get it? Or my doctor doesn't know about this. So can you, just as a clinician, can you talk a little bit about how people should ask for this and what's the best path for that?
Well, for C2N, as I said, they're on the market in 49 states. And all this approvals happened relatively recently, within the last year. So the company's figuring out their sales strategy, their marketing strategy. Basically, we have to educate doctors about the value of the test and where the test is available. Right now, there's a test that laboratories like Quest and LabCorp are offering, but they're not the same tests as the test that's been approved essentially by Medicare and through their mechanism, given an accreditation to the C2N test. So I think it's preferable to get the accredited test. We don't know how the Quest and LabCorp tests will be developed over time to a point where we can have confidence in them. But basically, there is a website. People can do a search on the test itself. It's called Precivity, Precivity, AD, A like an apple, D like in dog, number 2. So Precivity AD2 is one test that C2N is offering. And if you do a Google on that, the website will help you navigate locations in your area where the test is being available. And what we want to do is educate the doctors about how the test is administered, get the right supplies so that your local doctor ultimately will be able to order the test, and I'm sure they will.
Thank you for that. Which brings me to, why would you want to know? And we frequently talk about that as well. Probably many of you watching us today saw the last Alzheimer's patient on CNN. It was broadcast about a month ago. And some of our investigators that we fund their work was featured in that documentary. But there are proven steps toward prevention. So can you illuminate that for people, Howard?
Sure. When people come into the office, when patients come into the office and they have memory complaints, they deserve a diagnostic evaluation because sometimes memory complaints can be due to B12 deficiency, which is very common in the elderly, or thyroid disorders, which are very common in the elderly, or multiple little strokes, things like that, depression, polypharmacy, the effect of medications on memory. So people should go through an evaluation, a standardized kind of evaluation from your doctor. And then what we want to do is say, document that there is significant memory problems and cognitive problems. And once that's documented, again, we want to try to figure out what the cause is. And most of the time, we can say that the cause will be Alzheimer's disease. So that's kind of where the question, why would I want to know, if in the past, people would say, why would I want to know if there's nothing I can do about it? But the world has changed now because we have the first disease-modifying agents on the market. And it's certainly looking like the earlier that we intervene in treating people, the more the benefit is of these drugs. So it really comes down to the decision to treat, which is common for almost every disease. Think about cancer. Someone comes in with a cancer, it might be a deadly cancer. Does the patient really want to get that chemotherapy, which has so many side effects, for a very modest increase in their life expectancy, let's say? So every discussion about treatment involves what we call shared decision-making between the doctor and the patient, and usually the family, the husband, the wife, the spouse, to describe the possible benefit of the treatment, potential side effects and what they are of the treatment, the burden of the treatment, because these are a few infusions right now. And then together, the family can just express their wishes for what they want to do. I think that's very important, that shared decision-making. So people will come in because they're not knowledgeable about the potential benefit. But I think once we describe the benefit in a way that's understandable, I think people often say, yes, that could be very valuable. For example, from the Lilly trial with donanemab, it seems that about 50% of people who take the drug and get rid of the amyloid from their brain actually have a stabilization of their disease for about 1 year, whereas only 29% of people who took placebo had that stabilization. So one could ask themselves, doctor could express it, is, getting 1 year of stabilization at the level where you are, where you're still functioning in the mild cognitive impairment stage, and still able to recognize and enjoy and name your grandchildren, is 1 year extra life like that worth it to you, considering the potential side effects of these drugs, which in about 1 or 2% of patients can be very serious, but in the majority of patients, serious adverse events, which are about 20%, are reversible and manageable by taking patients, stopping the medication for a while, letting these side effects of what are called amyloid-related neuroimaging events, that those can be resolved by themselves, and patients can consider restarting the medication if they want. So we have a way of managing these what are called ARIA, amyloid-related imaging abnormalities. We have a way of protocols for managing the side effects that occur, which in most people are not serious. I mean, they're classified as serious by the FDA requirements, but in clinical practice, they're generally manageable.
And so, Howard, as we look back on the last 6 months of the year, what are you most excited about? What do you? I know you read widely, so what's got you up at night thinking, "Wow, that's really interesting or very cool"?
Well, I think one thing is we're seeing evolution of these anti-amyloid therapies now that we kind of know it looks like they're efficacious. So I think the next thing that will happen with these therapies will be home administration through sort of an EpiPen, the way insulin is given. I think that'll be a big breakthrough for individuals to be able to administer it themselves without having to go to an infusion center. I think it'll dramatically reduce the cost of these drugs, which is a burden to individuals through copays and to society through insurers like Medicare and even employer-based insurance. I think that we're going to see new versions of these drugs that are potentially more efficacious, drugs that help, chemistry that helps the drugs get across the blood-brain barrier into the brain in a more effective way. So I think we're going to see follow-ons, potentially even maybe not in the next year or two, but potentially some results of follow-ons of drugs that are pills that affect this pathway. So instead of having to go for infusions or taking an injectable, people may be able to take pills that kind of have the same effect in preventing plaques from developing and growing, and maybe even dissolving the plaques that are part of the brain. So there's that. And then there's all this 75% of other pathways, a lot of really exciting clinical trials on reducing the inflammation that contributes to clinical symptoms and the progression of the disease. Vascular components, we're seeing small companies that have programs against the vascular component of amyloid disease, particularly people that have amyloid deposition in their blood vessels. These people seem to have a faster progression of the illness. So companies like Therini that we're invested in have a treatment, an infusion product for helping to reduce the vascular component of the disease. We have drugs that are neuroprotective, so that regardless of sort of the mechanism that's causing the amyloid plaques and causing the neurodegeneration, these drugs protect cells by helping to stimulate the pathways that cells have inherently to promote survival and prevent cellular death. And that could have wide utilization in not only Alzheimer's, but in other neurodegenerative diseases. A company called Pharmatrophix that we've been investing in for many years. So I could go on. There's many exciting possibilities that are in development now. As I mentioned earlier, 75% of the approximately 120 drugs in development now are non-amyloid, non-tau drugs.
So are you ready for a little bit of Q&A with our audience?
I hope they don't ask me hard questions.
So I just want to remind everybody, for those of you who'd like to ask a question, you can put it in the Q&A at the bottom of your screen. And we'll start off with, well, this actually is sort of a follow-on to what you were just saying, Howard. What are some of the most promising targets for drugs right now, aside from amyloid, and how far advanced are these drugs?
Well, I think the drugs that are trying to calm down the inflammatory cells called microglia are gaining a lot of interest now. There's some clinical trials. A company called Elector has an interesting drug for that purpose, and we're funding companies like MindImmune and others that also have a target to manage. I mentioned Coya Therapeutics to manage the inflammatory component. I think that's very exciting. I mentioned Therini on the vascular side. We have drugs that are trying to attack the oxidative damage that happens in all of our cells every day. The oxidative just living on Earth, as we breathe oxygen, we get oxygen-free radicals that damage proteins and cause them to be malfunctioning and become toxic. And so trying to better manage the excess oxidation that occurs because as we age, we're not managing oxidative free radicals as well. And a company called Treeway, based out of the Netherlands, has we're helping them to develop an oxygen free radical a free radical scavenger that will help to remove these oxidative free radicals that are damaging proteins in the brain. And we're very excited about that because that particular drug called edaverone has been demonstrated to be effective in ALS, or amyotrophic lateral sclerosis. So in a way, it's a repurposing experiment, but we're just using a different version of the drug that's being used in ALS. The version that we're funding is actually an oral pill and not an intravenous pill. So those are just some of the ones. We're also starting to really literally translate the biology of aging. There's a group of drugs called senolytics, which attack what are called zombie cells. So there are cells that as they age, they die, but some cells refuse to die. We don't exactly know why, but they're called senescent cells. And by being there and maintaining themselves, they cause a lot of inflammation. So there's a repurposing of certain cancer drugs and other drugs to try to kill these senescent cells because we want to remove the inflammation. And we're funding Miranda Orr at Wake Forest University to do a clinical trial of a combination of a supplement called quercetin and a cancer drug called disinutab to remove the senescent cells from the brain and see if that will have a benefit in slowing the course of the disease. And it's one of our first endeavors in really literally translating the biology of aging through the development of a new class of drugs called senolytics.
Another question we have, Howard, is there a timeline for when the injectable anti-amyloids will come to market?
It's a great question, and I kind of think it's going to be sooner than later. But I don't have inside information on how soon. I just know that these pens are being developed, and I don't know how long the clinical trials will take, and then FDA approval and so on. So it's not going to be 10 years, but it may be as long as a couple of years or a year or two. But I really don't know.
Do you have a sense of when the C2N test will be approved in New York?
I wish I knew, because in my practice here, I'm very much a proponent of using these tests, and I think they'd be very beneficial for my patients. But because I practice in New York, I cannot order the test. And there are ways that I try to connect with doctors in other states like Florida, New Jersey, and Connecticut to help me, especially if patients have homes there. We can contact their doctors in other states and get the test done. So it's a little cumbersome, and I'm hoping New York will approve the test in New York in the very near future.
So we spent a little time before talking about donanemab. What's the next step for donanemab to be for the FDA approval and have it be moved into clinical practice?
Yeah, actually, that's a segue to something we haven't talked about, with just prevention. Mm-hmm. And we know through lifestyle changes, through the study called FINGER, which Mia Kivipelto has done, and Dean Ornish has done another study. We know that prevention works. So exercise, diet, cognitive stimulation, managing diabetes and hypertension work. What we're funding now is the leading anti-aging drug out there called metformin, diabetes drug that helps manage insulin resistance, which is a big problem as we age, especially in the brain, which is the most metabolically active organ in the brain. So and these cells in the brain are desperate for glucose, and if there's insulin resistance, then cells will undergo neurodegeneration. So we're combining metformin with Dr. Kivipelto's prevention program, just like we combine prevention, exercise, and diet and so on for heart disease. along with the statin. So that study is going on. And I think to answer your question about donanemab or the monoclonal antibodies to beta amyloid, we know that the disease starts about 20 years before people become symptomatic. So let's say the plaques are starting to develop in a person at 55, and they're not going to have symptoms till 75. We can do a brain scan, or now we could do the blood test to see if they have plaques. And then by identifying them, we can put people on preventative treatment to remove the plaques. And hopefully that would slow down or delay the onset of symptoms. We could even put people into a situation where they get the monoclonal antibody to beta amyloid. We monitor the removal and see when the brain is devoid of any amyloid plaques. And then maybe 3-4 years from then, we repeat the brain scan or the blood test to see if the plaque is reaccumulated. And if they do, give them another round. There has been one trial of this with a drug that failed, but it kind of set the stage for how we will do these prevention studies using therapeutics by identifying people in the community that have preclinical disease and giving them a therapeutic attacking the amyloid to see if that has clinical benefit. I'm sure that the study that was done called AHEAD, that study will be replicated with other antibodies that hopefully will be more efficacious and prove that we can prevent Alzheimer's disease, which I think is really the next most exciting thing that we're going to see in this new era.
Yeah. What are your thoughts on focused ultrasound that's being used to increase drug delivery across the blood-brain barrier?
Yeah, I'm a little biased because I used to use ultrasound in my laboratory. It's a common technique. You use what's called a sonicator. I know this focused ultrasound technology is a little different than what I was used to seeing to mash up brains so I could study them. I think they do have some positive clinical data. I just worry about, and I know they have safety data. I think we need more data to see the widespread use of the focused ultrasound. Let me put it that way.
Well said. What novel blood biomarkers are promising?
Yeah, well, certainly we can measure tau.
Mhm.
But the other thing that's coming on board, and we just had the guidelines come from the Alzheimer's Association and a couple of other organizations on the role of biomarkers in diagnosis. And so they had different tiers of diagnosis. Like, let's say we want to identify people that have a lot of inflammation in their brain so that we can give them these anti-inflammatories that are in development, or identify people that have a lot of inflammation in their brain and we want to get them into a clinical trial. So this very dense document that really went into this issue greatly identified the promising biomarkers. For example, GFAP, which tells us how a certain cell type in the brain is reacting if there's overreaction of that. A blood test called NFL, which tells us how the axons in the cell, the cables in the brain are degenerating. So that's another. So we have a neurodegenerative biomarkers, we have vascular biomarkers, we have inflammatory biomarkers, and others. And this document that just came out is summing up how these biomarkers can and should be used in clinical practice for diagnosing the various subtypes of Alzheimer's disease that are going to come on board, along with neuroimaging, I might add, which is what we think right now is probably getting an MRI is one of the more effective ways of looking at how much neurodegeneration there is in somebody's brain. So I think it's a really exciting time that I think that document will be useful in helping us to put together all of our thoughts of a really emerging area of biomarkers for the diagnosis and the prognosis and the clinical and the use of these diagnostics in clinical trials. We have a $100 million program called the Diagnostics Accelerator, which has made upwards of 70 investments in new diagnostics. And it's not just blood tests. We're looking at eye tests, looking at amyloid in the back of the retina, which, people imagine they could go to their annual eye exam and get a screening if they want to see if they have early Alzheimer's at a very low cost with no side effects. Or we also have digital biomarkers where we have something called the Speech Consortium, where we're gathering speech data on people, and then we're going to be able to characterize people that have problems with speech that are reflective of early Alzheimer's disease, because we know that that happens. Certain aspects of speech are abnormal in the very earliest stages of the disease. And there are other ways to use digital biomarkers that the Diagnostics Accelerator is investing in. One example is that we often ask people to draw a clock, and then it's a paper and pencil test, and the clinician kind of evaluates how the person draw a clock. Well, there's companies out there that have a digital version of that capture, I think, something like 1,000 data points on somebody's clock that's done on a specialized program on an iPad or something like that. So the ability to use digital technology to enhance our ability to detect aspects of cognitive impairment is another new frontier that's rapidly emerging.
Can you talk a little bit about Alzheimer's research looking at repurposing drugs, specifically metformin?
Sure. Well, what repurposing means is that you take a drug that's already on the market, where we know the safety characteristics of that drug, and we know the mechanism of action. And then we decide, well, would that mechanism action for, let's say, diabetes be useful for Alzheimer's disease? And then we would set up a clinical trial that would take that drug and test it in people with Alzheimer's disease. So metformin is a good example. About 7 or 8 years ago, we funded a clinical trial of metformin for people with mild cognitive impairment who also were at great risk for insulin resistance because of being overweight or obese. And we invested, I think, a few $1 million in that small study. And it showed some promising results. And so the investigator, Jose Luxinger up at Columbia, just got a much larger trial of metfinger for the treatment of Alzheimer's disease in people who are not diabetic but have certain characteristics that would enable them to be having increase the likelihood that these people have insulin resistance, and theoretically improving insulin resistance in their brain would slow neurodegeneration in that setting. And to give you an example of the value of repurposing there, that study actually stimulated a study of a drug that Novo Nordisk is currently investigating called semaglutide, or Ozempic, which also doesn't work directly on insulin resistance, but has an effect on the way cells use glucose and so on, and is also anti-inflammatory. And so, again, repurposing a diabetes drug called Ozempic, called semaglutide, for Alzheimer's disease, a very large clinical trial going on that really the company got confidence to do this, where it's a very expensive endeavor, through a trial that we funded at the Imperial College of London of the precursor to semaglutide, which was called liraglutide, which brand name Victoza, which showed very promising results. And Novo Nordisk was our partner in funding that study by Paul Edison as an academic investigator, repurposing diabetes drugs. And I think that's going to be a very promising way to translate the metabolic disorders of aging into therapeutics for Alzheimer's disease.
Switching gears a little bit, the Alzheimer's Association recently released new diagnostic criteria. Can you explain their recommendations around staging?
Sure. It's a very thick document, so I'll try to. We've always had a fairly clinical approach to making the diagnosis. It was not a very certain way of doing it. It had a lot of subjective elements in terms of the clinician's ability to do it. And that goes back probably 20, 25 years at least. I don't remember exactly when those first criteria were introduced. And then we started using through results based on, sorry, I'm blocking on the name of the study. It was based on I think it was the ADNI criteria, but using neuroimaging essentially as a way of making the diagnosis more precise. And in that case, there were clinical criteria, and then MRI criteria, and then PET scanning criteria. So we could look at neurodegeneration, the presence of amyloid or tau. But what's happening now is that the diagnosis has now become biologically driven. And, it's kind of like saying, you could sort of make a diagnosis of heart disease if someone was having chest pain. But now we can make a diagnosis of the risk factors that predict it, like cholesterol. And now we can even go so far as do neural imaging of the heart to see if someone actually has plaque in their coronary artery. So, it's a similar sort of evolution, but now what the new criteria are really biologically biomarker based, taking advantage of all these new biomarkers. And it's very timely because we've had such an explosion of research on biomarkers. Now we kind of have a way of thinking about how to use them. And basically, the preclinical stage of the disease really kind of completely relies on the biomarkers to say whether or not someone has the disease or not. I mean, there are clinical criteria, and then those are confirmed by the biomarkers. So that's like stage 1. And then stage 2, stage 3, stage 4, stage 5 go through more advanced clinical stages of the disease, continuing to use the biomarkers at different levels as defined in this detailed document, along with the clinical criteria. So stage 5 would be predominantly clinically driven severe dementia. Stage 4 is moderate dementia. Stage 3 is mild cognitive. Stage 3 would be mild dementia. Again, to make a diagnosis of Alzheimer's, mild dementia from Alzheimer's, using the biomarker strategy that's outlined in the document. Stage 2 would be mild cognitive impairment, the first symptomatic stage. And as I mentioned, stage 1 is the preclinical stage. But all of these now incorporating, I'm sorry, I'm repeating myself, the biomarkers that are outlined in great detail and usefully in this document.
So we have one more question for you, Howard, before we wrap up. We're going back to the blood test. Are blood tests able to be used for diagnosis now, or are they really considered a screening tool? Are they? Do they need to be FDA approved to confirm a diagnosis?
They're not a screening tool. It's not recommended that they be a screening tool. They're too expensive, and they don't meet the criteria of a screening tool, which would be inexpensive because you're going to want to apply these to large populations, and it would create a real. We talked before about shared decision making. To put this out in the community and people getting positive tests without a good system to counsel people. It's like HIV testing. We don't want HIV testing without counseling patients about what a positive test might mean for them. So that, same thing with the Alzheimer tests. But as a diagnostic instrument, I think they're very valuable, especially today when we have therapeutics attacking the amyloid, like the monoclonal antibodies. I think in that setting, the clinician can go through an algorithm where there's, the patient comes in with symptoms, they have memory loss, there's testing that's done. I already described the potentially reversible causes of memory impairment that have to be ruled out, and then going to a specific diagnosis of Alzheimer's or not through one of the blood tests. I think that will turn out to be very useful because it'll help people to get to a definitive diagnosis. It'll help people to decide whether or not they want to go into clinical trials, or at this point, these monoclonal antibodies are clinically available through prescription, and doctors can prescribe them for people who have the right criteria, including a positive blood test. And probably, we don't know yet, maybe a blood test confirmed by an imaging exam. I think that's one thing that is not exactly 100% clear yet, but it may turn out the blood test will obviate the need completely for the brain scan as a confirmatory test before people get the monoclonal antibodies. I think that's a little bit in a state of flux right now in terms of how we'll proceed.
Well, Howard, I want to thank you so much for joining me for this 45 minutes. Always wonderful to hear from you. And on a personal note, you as a clinician, I think you certainly give me a very hopeful outlook on what is happening within this field. And we promise that we'll come back in a couple of months and do another update on where we are in the field. But thank you all so much for your support of the ADDF. And we wish you all a very happy 4th of July holiday. Thank you.
Thank you.